Identifying bacteria is one of the most important tasks in microbiology. Among the many biochemical tests, a classic group known as the IMViC test is widely used to differentiate members of the Enterobacteriaceae family. In this video, we'll break down what the IMViC test is, how it is performed, and why it matters. IMViC is not a single test but a series of 4 biochemical tests. I. Indole test, M. Methyl Red test, V. Voges Proskauer test, C. Citrate utilization test. Together, these tests help microbiologists distinguish between closely related bacteria such as Escherichia coli and Enterobacter aerogenes. Let's start with the Indole test. This test checks whether the organism can break down the amino acid tryptophan to produce indole. The medium used is tryptone broth. After incubation, we add Kovacs reagent. A red or pink ring on top indicates a positive result, meaning indole is produced, as in E. coli. No color change means negative, as in Enterobacter. Next is the Methyl Red test, IMViC test. This test checks whether the bacteria perform mixed acid fermentation of glucose. The medium used is MRVP broth containing glucose. After incubation, we add a few drops of Methyl Red indicator. A red color means positive, showing strong acid production, E. coli. A yellow color means negative, indicating less stable acid production, Enterobacter. The third is the Voges Proskauer test, VP test. This detects whether the bacteria produce acetoin, a neutral end product of glucose fermentation. Again, MRVP broth is used. After incubation, add Barrett's reagents A and B, alpha-naphthol and potassium hydroxide. A red color after shaking indicates a positive VP test, seen in Enterobacter. No color change means negative, as in E. coli. Finally, the citrate utilization test determines whether the bacteria can use citrate as their sole carbon source. The medium is Simmons citrate agar, which contains citrate and bromothymol blue indicator. If the organism can use citrate, the medium turns blue, positive. If not, it remains green, negative. Enterobacter gives a positive result, while E. coli is negative. When we combine these 4 tests, a pattern emerges. E. coli, indole, plus, EMSAR plus, VP, citrate, Enterobacter, indole, EMSTER, VP, plus, citrate, plus. This IMViC profile makes identification faster and more reliable in clinical food and water microbiology. The IMViC test is simple and cost-effective, useful in distinguishing coliforms in water testing, widely applied in teaching labs, clinical microbiology, and environmental monitoring. In conclusion, the IMViC test is a powerful tool to identify and differentiate Enterobacteriaceae. By understanding each step, indole, methyl red, Voges Proskauer, and citrate, you gain a solid foundation in microbiological testing. Thank you for watching. If you enjoyed this explanation of the IMViC test, please like, share, and subscribe for more microbiology tutorials.